All right, we have successfully finished our toll-like receptors. And that means we get to talk about nod-like receptors. And oh my gosh, you see how there's a similarity in the way they're drawing this thing? There's a structural similarity and an evolutionary similarity between these proteins. Yeah, they're actually just like an extension of the same thing. So whereas humans have these 10 toll-like receptors, I believe the sea urchin has over 100 toll-like receptors, and so it can just respond to more different things. So instead of being able to make antibodies, it just makes a whole bunch of toll-like receptors, and it just keeps modifying those. It's kind of a slower version of an immune process, but you can get to roughly the same place. Yeah, they survive just fine. And nod-like proteins are also really widespread, and they go right back down to the base of the animal evolutionary tree. And so you can find these things, and they have always a couple of domains in there that are common. So the outside is called an LRR domain. That's the thing that's actually going to pick up the whatever it is that we're binding to, something like bacteria, for example, something that's come from them. Or this muramyl dipeptide. This is a component of the bacterial cell wall. And at the bottom of it, instead of that TIR domain, we're going to have a NOD domain. So that's why it's a NOD-like protein. And it's going to be related to, I believe, it's the fruit fly protein that is also called NOD, N-O-D. And then at the bottom of that thing, we're going to start clipping onto some very interesting proteins. So a CARD domain is called a caspase recruitment domain, and caspase is CASPASE. So caspase recruitment domain will recruit caspase proteins. Now, I'm guessing you may have already had cellular biology with Dr. Alam or whoever you took it with. And I'm guessing that in cellular biology, at some point, you would have run into caspases. These are some of the proteins that are involved in programmed cell death. And the way you activate these things and stick them together is with caspase recruitment domains. So a protein with a CARD domain on it, caspase recruitment domain, is serious business. And this can lead to a cell killing itself. And you'll see that a lot of the proteins involved in this process are going to have these names that are, I would describe them as totally metal. Yeah, like RIP2. They all are something like you'd find in a, I don't know, Disney Halloween graveyard. That kind of name, that just sort of right on the nose kind of name. So anyway, yeah, in their inactive form, they're just going to be in the cytoplasm. So remember, we've got our toll-like receptors that are either on the cell surface or are monitoring whatever that cell, like a phagocyte of some sort, has just eaten. And then these guys are floating around the cytoplasm. So these are a way to tell if there's something that's going on that is not at the surface and not in the endosome, is basically anywhere else in the cell. So that's good. Between NOD proteins and toll-like receptors, the cell has kind of got it covered. It's got its bases covered. And so our NOD protein is going to interact with RIP2, yeah. And it's actually going to be two copies of that. It's going to pair up just like the toll-like receptor did. And the thing that it binds to is what's actually going to join these together. It helps them to come together. And then the caspase recruitment domain is going to bind to RIP2. And it's going to cause it to unfold, change shape, and bring in a ubiquitin ligase. And so we don't have to remember which ubiquitin ligase. It's this XIAP. But it is going to lead to polyubiquitin formation. So we're going to make a multi-ubiquitin chain once again. And that multi-ubiquitin chain is going to be made and attached to this guy, which is our little TAC protein, TAC1 complex, which is going to be phosphorylated. And that has to get phosphorylated somewhere else. But once it is phosphorylated by another process, so this is going to require some outside intervention, if you like, or perhaps activation of the toll-like receptors could lead to just a second way that we can amplify that signal. So if we have activated TAC1 from one of the toll-like receptors activating it, then it can also come over here. And yeah, if there's anything that the NOD proteins have picked up, we can make a bigger signal and make a bigger response. And so that response is actually going to look pretty much the same. And so we're going to bind this IKK complex, which is going to be NEMO, which I think is IKK gamma, if I remember right, and alpha and beta, but just call it the IKK complex. That's fine. And so TAC1 is going to put that phosphate onto the IKK complex, and we don't care where at this level. And that is going to cause the IKK complex to put a phosphate onto I kappa B, which they haven't even drawn here, but that's totally what it does because that's its function. And that's going to make NF kappa B be free because I kappa B is going to get destroyed once it gets phosphorylated. NF kappa B, nuclear factor kappa B, can then go into the nucleus. And when I'm saying kappa B, I'm just pronouncing the lowercase Greek letter K that's here, which is a kappa. Yeah, as part of the word. And so that's how people call this thing, NF kappa B. Okay, so there are different domains that you find in these NOD-like receptors, and they're actually pretty widespread. And so, for example, some of these things will have tier domains, so they can actually interact with toll-like receptors. So things like MyD88 and MAL and TRIF and TRAM and all the TLRs have those tier domains. You'll get these caspase recruitment domains that you can find on caspase 1, so that's why it's called caspase recruitment, but also on things like RIP2, RIG1, MDA5, MAVs. These are things involved in a different intracellular immunity system that we cover pretty heavily in virology class, and I think we're just going to lightly cover here, perhaps. Also in pyrin proteins, and there are a bunch of proteins that have a thing called a death domain, abbreviated as DD, yeah, which can eventually, down the road, lead to a cell killing itself. And then the death effector domain is another domain that is going to be found attached to these NOD-like receptors. And the death effector domain, yeah, is dead, DED. That's going to be found on some of the caspases that will actually end up killing the cell, like caspase 8 and 10. Okay, so how do these things react to the world? So the idea is that these NOD-like receptor proteins, that's what NLRP, so they're related to NOD, or they have that NOD domain in them. NOD-like receptor proteins are going to react to various kinds of cell damage, and something bad that's happening inside of the cytoplasm. So if there's something that happens, a bacterial toxin comes in and causes your cell to just lose its ionic gradient. So cells spend a lot of their day and a lot of energy maintaining ionic gradients, keeping more of one ion on the inside versus the outside through active transport. This builds up a membrane potential, among other things. And you have this to some extent in all living cells. But yeah, if that potential disappears, then, oh my gosh, our NOD-like receptor protein 3 will actually have a pyrin domain that will interact with the card domains on procaspase 1, and we will then end up causing caspase activation. So yeah, that's just how it's going to happen. And as part of this activation, active caspase 1 is going to make some really serious stuff. So interleukin 18 and interleukin 1 beta are really, really, really strong cytokines. And they're actually made, once again, in a safe form. So they have this extra protein attached to them. But when we cut it off, that's what caspase does. It's a little protein cutting enzyme. It's going to release this little part, which is the part that we call interleukin 1 beta. Or interleukin 18. And both of these are very strong pro-inflammatory factors. And so these will call in other cells that will punish and destroy this cell or anything else in the area, because that's what needs to happen at that time. So here's a little model of what this actually looks like. So caspase 1, as shown down here, it's a little polymer here. It's our procaspase 1. And this is our NOD-like receptor protein 3. It's actually going to link together. You're going to get 7 copies of this thing, and you can see them all right here in the little picture. 1, 2, 3, 4, 5, 6, 7. And yeah, together those are going to bind to their little adapter proteins. And this is our pyrin domain at the bottom that's going to mediate that binding. And we're going to activate some stuff. And we're going to start making interleukin 1 beta, among other things. All right. Our other proteins, and these are involved very heavily in antivirus responses, are going to be RIG1 and MDA5, or some people will call that RIG-I. It's a Roman numeral 1. And it's a retinoic acid inducible gene 1. And MDA is muscular dystrophy associated gene number 5. Back when we didn't know exactly what these things did, this is the best we could do, was to give them just some kind of generic name. It turns out that RIG1 and MDA5 are pretty much the same gene. They're like 2 copies of the same thing. They're set off by a lot of the same signals. There's probably a little bit of difference between them, but it may just be whichever one is on hand that gets to activate. And so what happens is that RIG1 and MDA5 both have these caspase recruitment domains on them, but they are folded up, just like you would fold up a pocket knife if you don't want it to accidentally stab you in your pocket. And when it binds to a little piece of a virus, and so somehow they're recognizing some part of a virus. We think it is probably the cap structure if a virus has been a bit sloppy. Then this will open up and it'll actually run along the virus, and they'll get a bunch of these things together. And so they'll pick up a protein called TRIM25, and TRIM25 is another one that we cover in virology. It's going to be one of the factors that can stop a variety of viruses. And once we've got TRIM25 there, we're going to add ubiquitin onto this. So a nice little polyubiquitin chain is added. That allows the complex of RIG1 or MDA5 and TRIM25 and polyubiquitin to associate with MAVS, which is the mitochondrial antiviral signaling protein. And yeah, the ubiquitin is polyubiquitin attached at lysine number 63, but that's a detail that I would say you don't need in order to make your life better at this stage. So you can leave out the lysine 63 if you want, but it is true, and it is that is where it's attached. And that's why this particular polyubiquitin will bind to MAVS, because it is lysine 63 polyubiquitin on a RIG1 or MDA5 protein. That together is what MAVS is looking for, or on a TRIM25. Or I think TRIM25 has actually made the ubiquitin chain. And then once we have that together, and MAVS is actually bound to the opened up helicase domain via the card domains, that's what's actually binding to MAVS, then it's going to call in TRAFs. TRAFs are going to make a polyubiquitin signal, and that polyubiquitin is then going to activate the IKK complex, which will then phosphorylate I-kappa B, which will then leave NF-kappa B so it can go into the nucleus, or it will phosphorylate TBK1, which will then phosphorylate IRF3, which will then go to the nucleus and make changes. So I would say we can leave out these steps right here. We just need to know that the RIG1 or MDA5 is directly binding to virus RNA, and when it opens up, it exposes its card domains that bind to the card domain at the top of MAVS, the mitochondrial antiviral signaling gene. And then our card domains, which are now stuck together, are going to recruit TRIM25, which is a ubiquitin ligase, and is going to add little polyubiquitin chains on here, which will then provoke a response that will either make IRF3 go to the nucleus, or will make NF-kappa B go to the nucleus. So those are the outcomes. And we know this is important in antiviral defenses because, one, if you knock it out, cells become incredibly susceptible to infection. And two, there are a lot of viruses, including the coronavirus, that will have 4 or 5 different proteins specifically dedicated to shutting down every part of this pathway. So coronaviruses just really don't like this pathway and are really trying to rein it in, basically. Yeah, to make just the right level of immune response. Not too much, not too little, just like Goldilocks. Okay, so that's a good place to pause here. So we've talked about our RIG1-like receptors. Yeah, RIG1-like receptors. We can forget MDA5 if you want. We can just call it the RIG1-like receptors and go through it that way. And we've got our, let's see, our NOD-like receptor protein 3, which have this extra NOD domain down in here. Those are the other ones that we should learn in this section. And these are both parts of the innate immune system that can lead to a cell making an immune response to a generic form of danger. So a virus with something wrong at the end of it, for example. Yeah, or anything that produces an RNA that has something wrong at the end, an extra triphosphate where there shouldn't be. That's enough to set off one of these things. So thank you very much. That's enough for this section.